Sayın Dilan Köksal Öztürk hocamıza sunumu için çok teşekkür ediyoruz. Şimdi sırada Sayın Berin Karaman Mayack hocamızın hazırlamış olduğu video sunumu vardır.
Hello everyone, this is BERİN KARAMAN MAYACK speaking. Today I would like to talk about the in silico work done on
Hocam yapmak istiyorsanız tabii hocam hemen mikrofonu açıyorum.
Teşekkür ederim. Sanırım yanlış bir anlaşılma olmuştu. Çok sağ olun hocam. Hemen ekranımı paylaşıp...
O zaman video paylaşımını koymamızı istiyor hocalarımız da o yüzden bu şekilde düşündük. Buyurun hocam.
Anladım hocam. Çok teşekkür ederim. Hemen sunumuma geçiyorum. Görebiliyoruz değil mi?
Evet hocam görüyoruz. Buyurun.
İngilizce hazırlamıştım sunumumu. Hello everyone, this is BERİN KARAMAN MAYACK speaking. Today I would like to talk about a specific in silico approach that we conducted on microRNA prelate7 and protein Lin28 interaction. And the aim of this study is to use computer-aided drug discovery approaches to identify novel small molecule inhibitors of this interaction. So when we looked at the prelate7 microRNA biogenesis, we know that microRNAs are small non-coding RNAs and they regulate the gene expression. And specifically, the prelate7, specifically the microRNA LED7 is regulating the gene expression and the proliferation of some genes. And when we looked at the biogenesis of microRNA LED7, we see that initially the prelate7 is in the nucleus found as the primary LED7, as transcribed as primary LED7, and then it is cleaved by the DROSHA enzyme, and this precursor LED7 is transferred into the cytoplasm. And in the cytoplasm, there's another enzyme which is called dicer, and dicer is transforming this precursor LED7 into mature LED7. And mature LED7 is quite important because it repressed the translation of some oncogenes. So we see a gene silencing through this microRNA LED7, which therefore is found to be quite important in the development of many cancer cells. And what happens is that when Lin28 is bound to the either the primary LED7 or the prelate7, we are basically blocking this process where the mature LED7 we have at the end. Therefore, when we have the Lin28 binding to our microRNA LED7, we are actually blocking this gene silencing of the oncogenes. And therefore, it is thought as that the Lin28 may be a good candidate for predicting the cancer development, and therefore it can be a good anti-cancer therapeutic target. Therefore, we in our work focused on identifying small molecule inhibitors that can actually inhibit the interactions between the Lin28 protein, where you can see the two different domains of this protein in light blue and in green in here, which interacts with our microRNA LED7. And there are two domains that are making these interactions with our microRNA LED7, which are the CSD, which is the cold shock domain, and the ZKD, which is the zinc knuckle domain. And in the literature, when we looked at the recent studies, they found out that there's a relatively higher affinity between this cold shock domain and the microRNA in comparison to the zinc knuckle domain interaction with the RNA. Therefore, it is suggested that it is more favorable to identify small molecule inhibitors for the binding onto the cold shock domain. And in our work, we are aiming to focus on these small molecules that can bind into the same region where the microRNA is binding to the CSD domain. You are in here seeing an overall workflow that is implemented in this study. And what we have done is we actually designed a workflow that consists of both ligand-based and protein-based virtual screenings to identify novel molecules. In the first step, we prepared a human Lin28 protein, and then we also prepared a commercial library where we will be looking into if there will be any compound that have a good binding affinity to our CSD domain in Lin28. And in order to judge which compounds will have a good binding affinity, we conducted a hierarchical virtual screening approach, which starts with a ligand-based virtual screening, and we use fingerprint-based virtual screening in that process. And then we conducted a structure-based virtual screening, which is done by molecular ducking, and the molecular ducking is followed by protein ligand interaction-based filtering. So when we looked step by step of these experiments, the first one was taking the human Lin28 protein in complex with our microRNA pre-LED7 in here. And we got this protein microRNA complex from the protein data bank, and we prepared this complex using the protein preparation wizard module, which is implemented in the Schrödinger suite. What we have done in short is we added the missing hydrogen atoms to our crystal structure. We corrected the bond orders and assigned the charges to the atoms. And we optimized the hydrogen bonding network, and we sampled the water molecules that are present in this complex structure. And finally, we minimized the whole complex to get the most stable energy, minimized conformation. In the second step, what we have done is that we use this commercial virtual screening library, which is Cambridge, which is open to everyone. And when you identify any molecule through this virtual screening library, you can actually go and purchase that compound for your biological testing. And what we've done, we downloaded all these compounds, more than 1.5 million compounds, and we prepared using the ligand preparation module, which is called LigPrep, and which is implemented in the Schrödinger program again. And what we have done is we prepared our ligands at pH 7 plus and minus 2, and we we generated different ionization and tautomeric states of these ligands. We also considered the stereochemistry of these compounds and generated different ring conformations. As we already know, some of the compounds in the biological testing and in vitro testing, they are so-called frequent hits, which means that these compounds are giving us false positive results in these 10 assays. And therefore, we use this already reported pains as pan-assay interference compounds and remove these compounds from our databases. In the next step, what we have focused on trying to make our library more enrich it with the drug-like compounds. Therefore, we used rapid elimination of civil rules to remove the compounds that are less likely to be a lead compound. And in the final step, what we have done, we further filtered our database to enrich it by just molecules that are more drug-like. We used molecular weight, log p-values, number of hydrogen bond donors, acceptors, and such. And we filtered our compounds with this thresholds. And then in the next step, now that our protein is prepared, our database is prepared, we could actually start our ligand-based virtual screening studies. And as a first step, we use fingerprint-based virtual screening. And what we used MolPrint 2D representation of these chemical compounds in this work. So what we have done, we first collected the non-Lin28 inhibitors that are specifically shown in the literature that are binding or targeting to the cold shock domain. And then we generated the fingerprint representation of these chemicals as shown in such as an example, and that the computer can understand. And we also did the same thing for all the molecules that are presented or prepared in the database. So we also generated their MolPrint 2D finger representations. Then what we have done, we have compared the fingerprints of these non-inhibitors with the fingerprints of the molecules in the prepared database. And we use to compare the similarity of these compounds, fingerprints, we use Buser similarity metric. And we told basically the computer that if the compound has 85% similarity with the non-molecule, then we will select you. And we have compared all the molecules with the known inhibitors for the selection. And this was our first filtering. The compounds that met these criteria were subjected in the next step to molecular ducking. And for molecular ducking, we use the glide ducking tool implemented in Schrödinger software. So we also followed a hierarchical ducking experiment in here. So what we have done...
Hocam toparlayabilir miyiz lütfen?
Yes. And what we have done, we use high throughput virtual screening scoring function and standard precision scoring function to filter these compounds. And then at the last step, what we have done, we looked at the crystal structure and see how our microRNA in the real life scenario making interactions with the CST domain. And we use this hydrogen bond interactions to filter our poses, the molecules further. And only the compounds that have these interactions seen in the crystal structure were selected for the biological testing. And with that, I would like to thank you for your attention and hope to hear your questions. Thank you.